So I'm noticing a couple things. Number one, it's a long, skinny room and little baby slides. So I don't know what people can see in the back. I know that everybody's going to have the slides online, so please use them at will. The other thing is that I have now 32 minutes to talk about all of outcome measures and biomarkers, which, at least by my reckoning, is sort of a major topic in clinical trial design. So I obviously can't do justice to the topic in the time I have. So I'm going to try to focus on a few points, and I'm here all afternoon with office hours and happy to talk about individual projects. Just grab me at any time. So, disclosures. And so the word biomarker is something that is used frequently in clinical trials, and its actual definition is so broad that it encompasses almost everything we do in clinical medicine and experimental patient-based research. If you look at it in my Bible, Wikipedia says it indicates a measurable indicator of some biological state or condition. And the NIH definition is that it's an objectively measured and evaluated outcome that serves as an indicator of normal biological processes, pathogenic processes, pharmacologic responses, target engagement, or therapeutic intervention. And so you can see that this basically, biomarker covers about anything that we record in the context of a clinical trial, including all of the endpoints that form the basis of phase 3 trials, which are clinically relevant endpoints, which I'll talk about a little bit later. However, there's a structure in terms of what kinds of biomarkers you might think of using at different stages of study. And here's a table that refers to what kinds of endpoints you might want to use in different circumstances. So, on the left are the things you want to know when you're measuring something. And so, starting with target engagement, certainly if you have an intervention, you want to know that the intervention actually goes where it's supposed to go and has its intended effect on a target. And this is certainly an important early stage outcome. It's not likely to be an outcome or a biomarker that would serve as the basis of a FDA approval, but it's certainly something you want to know. And sadly, it's something that even in phase 3 trials, many areas of neurology never get to this category of biomarker throughout the entire stage of clinical development. And so you can measure this in a whole host of ways. You can measure this with fluids, you can measure this with imaging, PET, MRI, you can measure it with physiology. There are a whole host of different modalities you can use for this purpose. Pharmacokinetics is a critical aspect of therapeutic development. I don't know that I have time to do justice to this at all, but clearly you want to know where the drug goes in the body, how it's excreted, by what mechanism, and how long it takes for all that to happen. Pharmacodynamic markers include target engagement markers. So this table is a little bit misleading in that sense. So a pharmacodynamic marker is a marker that tells you something about the biologic process that underlies the disease or the intended molecular effect of the therapeutic. And pharmacodynamic markers are increasingly used in essentially all phases of clinical development. The early phase 1 development often includes in human beings some measure of the effect of the molecule on the organism in some fashion, although not the kind of effect that would serve as a phase 3 outcome. And so, again, they can be, a pharmacodynamic marker can be a fluid, an image, or a physiological test, or a late phase functional outcome as well. Markers are also clearly necessary to make the diagnosis, and so some of our diseases can be made on the basis of fluid markers, myasthenia and acetylcholine receptor antibodies. Some of them can be made on the basis of imaging, and some of them actually still have no diagnosis-related markers that are validated. And the disease I work on, ALS, is an example of that, even though I'll show you examples of things that may serve as that in the future. And so those diagnosis-related markers or stratification markers are important in clinical trials, obviously, to enroll the right population, to the extent that you can have a marker that distinguishes one patient with a disease from another in a way that's important with respect to the mechanisms of action of your drug. Those can be very important in stratification and ensuring that your groups are equalized. And again, an example of this is a disease that has both a sporadic incidence as well as a genetic influence. Knowledge about the genetic status of your subjects will allow you to equally in both your control groups and your treatment groups. Outcome measures, sort of writ in large in terms of late phase design, are also biomarkers, and they can be clinically relevant outcomes such as survival or functional rating scales. They can be functional outcomes such as strength or walking speed or intellectual function on a psychometric test. And these are getting closer to the biomarkers that one needs to bring something to approval in a phase 3 study. But not all disease outcome-related markers have achieved the level of qualification or validation that are necessary to be phase 3 outcomes, and we'll talk a bit about that. And obviously safety is a biomarker as well and is relevant in all phases of clinical trials. So this slide just represents that when you use which kinds of markers depends on what you're interested in. In terms of mechanism of action, this table indicates that that's an early phase question, and ideally that's true, but you'll find that mechanism of action may not be completely worked out by the time you start bringing your agent to people, and even in later phase studies, the proof that the mechanism of action of your intended therapeutic is actually correct is sometimes a middle or late phase question. Again, pharmacokinetics is an issue that should be solved earlier rather than later in clinical development. You can see at the top, we go from preclinical studies to post-marketing studies. And so pharmacokinetics is certainly addressed early, but issues certainly arise that need to be addressed later in development as well. Disease stratification issues are often ones for late phase trials because you need to make sure that your pivotal trial is appropriately randomized and stratified. And then disease progression markers are presumably the things that you're looking at ultimately in the later phases of investigation, both for approval endpoints and also for understanding the spectrum of effects that your agent may have. So a good chunk of the non-phase 3 primary endpoints are pharmacodynamic markers. And again, they can be markers of target engagement, target activation, or they can be markers of the intrinsic biological effects of the disease itself that you're hoping to modify. And they can be fluid, so that, you can look at enzymatic systems. More and more, we look at changes in gene expression through the products, either RNA or proteins. Or they can be specific imaging modalities, and I'll give you some examples of those. In neuromuscular disease, we use physiology as measures of the ability of your therapy to impact a given target. Physiology is not usually used as a approvable endpoint, but it's certainly a downstream marker that impacts that usually has more of a implication for the organism than simply an enzymatic target. And then and these things are used in a variety of ways depending on the phase of your study. So they're used to just give you yes/no answers. Does this drug hit its target? But they're also used for dose ascertainment as well as safety. So a couple of examples, starting with fluids and moving to other kinds of markers. I'm going to be ALS heavy, but not ALS exclusive. So as many of you know, genetic manipulation is in the forefront of clinical trial research these days. and antisense oligonucleotides are an attractive therapeutic modality, and in ALS, about 20% of genetic ALS is due to a mutation in the SOD1 gene. And so an antisense oligonucleotide, so an oligonucleotide that binds to RNA and presumably reduces the abnormal protein levels as well as normal protein levels, has been developed over the course of the last 7 or 8 years and is now in clinical trials in ALS. And the particular antisense against SOD1 is a blocking antisense, so it reduces SOD1 protein and has been shown to, by this mechanism, prolong survival of animal models that have the human SOD1 mouse, human SOD1 gene transgenically present in the experimental animal. And so the primary outcome measure of a middle phase SOD1 antisense trial in humans with ALS is SOD1 protein levels in the CSF. And the preliminary data for that comes from rats, which shows that in the SOD1 transgenic rat, the antisense molecule can reduce protein levels in the CSF in the experimental animal. And to the extent that you reduce CSF SOD1, you can show that it reduces brain SOD1. So that's what these two slides show. That's the experimental defense for using it as a biomarker in humans. An interesting aspect about this particular biomarker, which actually is neither a good nor a bad thing, but is important to know before you start using a marker, is how does that marker change over time with disease? And so these are SOD1 levels in the CSF of ALS patients over time without treatment. And you can see that the levels don't actually change. So that from the beginning of the disease to later phases of the disease, the levels are the same. So this marker is not going to be a disease progression marker. But it certainly could be a target engagement marker, and you would expect, to the extent that your RNA is blocked, the protein that it produces should be reduced. And that's the primary outcome measure of a middle phase trial now in progress. It's sponsored by Biogen. And so this is not a target engagement marker. The jump from reducing SOD1 to impacting disease is an experimental jump. But there's no implication here that if this study is successful, that forms a clear link to a clinically beneficial result. That would be the next study. A more closely tied disease progression marker has to do with inflammation. And so this is again ALS, looking at regulatory T cell levels and FOXP3, which is a T cell expressed protein, showing that in ALS patients, aggressive ALS, fast progressing ALS patients have a lower level of regulatory T cells than either normal subjects or slower progressors, and that FOXP3 is reduced. Now, here's an example of a phenomenon related to the disease that you could choose to target with your drug. And so in that case, it would be both a pharmacodynamic marker as well as that was relevant to disease progression, but also a marker that would be relevant to target engagement. And then finally, on the fluid side, again with respect to ALS, phosphorylated neurofilament heavy chain is a marker of axon degeneration and is currently being developed in ALS as a diagnostic marker and potentially both a prognostic and a target for intervention. On the left is a scatter plot showing that the level of phosphorylated neurofilament is related to disease progression, so that the more neurofilament you have, the shorter your disease progression is likely to be. And if you stratify patients according to disease progression, according to levels of neurofilament, patients with higher levels of neurofilament will progress more quickly than patients with lower levels. Now, similarly to SOD1, this is a marker that also doesn't change over time. So this is not a disease progression marker. It's a prediction marker in terms of how fast your disease may progress, but the level of NFH doesn't progress over time. So 3 types of fluids that can be related either to just looking at whether your drug hits a target or potentially markers of underlying disease activity. These are PET scans looking at with a marker called PBR28, which binds to activated microglia. And on the left-hand side are patients with ALS, and on the right-hand side are normal controls. And you can see that there's uptake in brain areas in ALS patients that don't show up in controls, suggesting that this marker is able to detect an abnormality that is not seen in non-disease patients. Now, this could be used as a diagnosis marker. It potentially could be used as a target engagement marker if you have a drug that you intend to reduce microglial activation. This would be a marker that would say you hit your target. Interestingly, although I don't have a slide over time, this is yet another marker that doesn't seem to change over time with ALS. So none of these actually are useful in assessing progression, but they're useful in assessing your ability to impact the target. And potentially, even though under normal circumstances you don't see a change over time, if you can induce one with your drug, then the experimental question would be, is there a clinical effect? So all of these are markers that are increasingly important to look at, but none of them have a clear implication that they relate to a clinical outcome. Other fields are doing much better with regard to sort of pharmacodynamic markers, and in particular, multiple sclerosis has been using gadolinium-enhanced lesions as a marker for many years. And this brings up the topic of surrogate endpoints. So we talked about pharmacodynamic markers, we talked about clinical endpoints. A surrogate endpoint is an endpoint that is not directly clinically relevant, but is presumed or shown to very closely relate to an important clinical outcome. And so that is how MRI lesions have been used in terminal development in MS for a number of years. And here's an example of oral fumarate in a phase 2 study. So this is a fairly small MS study, about 260 patients, and the outcome is number of new gadolinium-enhancing lesions. And in this study, where subjects were studied over a fairly short period of time, a clear reduction in lesions was noticed over the course of evaluating a relatively small number of patients for an MS study. And so this is not a clinically relevant outcome. This is a marker that is intended to predict that. And this is a middle phase endpoint that then led to a phase 3 study with an outcome that people would think as directly clinically relevant, which is relapse rate. And so this is a much larger study, 1,200 patients, showing that the clinically relevant outcome was predicted by the changes in MRI. And so that's what you would hope for a pharmacodynamic endpoint, that not only is it amenable to your treatment, but also that as you learn more about your disease, you're more and more confident that that endpoint predicts something that is clearly relevant and that can be used for approval by the FDA. So that's just a brief sort of pass through different ways that non-clinically relevant laboratory measures can be used in middle stage development. In later stage, we end up being more interested often in functional markers and in markers that have clinical relevance, and I'll define that as we go. And so in the neuromuscular world, there's a whole host of functional endpoints, and they can be related to strength, to pulmonary function in a variety of diseases, to walking, to fine motor movement. There's a whole host of them. And the choice of which one you pick depends on your phase of development. But even once you pick an endpoint, it's how you measure it and how you statistically treat it turns out to be very important, not only to the result you get, but to how meaningful that is to regulators later on. And so strength is a good example just because it's an intuitively simple concept that we measure in a variety of ways in both clinical and experimental neurology. So as neurologists, we measure strength all the time. We use the Mrc Rating Scale, which is an incredibly poorly designed qualitative measure. And that, at least even within the last decade or so, is still a way that people are using strength evaluation in clinical trials. And I'll show you how much of a problem that can be. And then, even once you measure it in a more systematic and quantitative way, how you evaluate it actually can determine whether whether changes in strength are a sensitive measure of your drug intervention or an insensitive measure. So this slide lists the different ways you can measure strength from qualitative to quantitative, and just one description of how bad a measure of qualitative strength is I think shown here. On the top, looking at biceps, grades 1 through 5, which are 0 through 5, which are no strength to normal strength, are represented in the top bar. And you can see that as you relate that to actual force, which is represented in newtons on the bottom bar, you can see that virtually all of the dynamic range of your strength exam is reflected in about a fifth of your ability to actually exert power in a given muscle. So you can go from 1 to 4 with a minimal change in strength, but then the entire rest of the dynamic range is virtually not represented at all. And so people who move to quantitative measurements of strength, that's not a big surprise. And at least in ALS, we've learned a lot of things, which is that although ALS is thought to be fairly randomly a random hit disease, it can affect anybody anywhere. It turns out that it's quite reliable, that certain muscles change faster than others, that it doesn't really matter which ones they are. But this graph shows that in a very large data set, certain muscle groups change much faster than other muscle groups, and that has implications for which muscle groups you might want to choose if you're going to restrict your data set and measure something in a more efficient way. But if you average them all, this is two large ALS studies over time. If you look at the two bottom graphs, those are average strengths over time. Not surprisingly, strength goes down. But if you average them all out, you get a measure that's quite variable, that requires hundreds and hundreds of patients per group to see a change over time. So even though it's a quantitative way of assessing motor strength, it's not a terribly sensitive one. And so over the last year or so, statisticians at Biogen and myself and some others have been looking at different ways of evaluating motor strength. And this is changing a continuous quantitative measure to a time to failure endpoint, which is sort of an important concept because you can measure endpoints either continuously or discretely. And so what we simply did is looked at the beginning of a study how many of the muscles were actually recorded as zero strength, and then of those non-zero strength muscles, how many of them reached a zero strength endpoint at the end of the study. And you can show these are individual graphs of individual muscles and their proportion of their probability of reaching a zero strength endpoint at the end of a 12-month period. And if you create an endpoint that actually says, okay, of all of the non-zero muscles, I'm going to take as my endpoint the development of a new zero muscle, so a time to the first new zero. And you look at the number of occurrences of that as compared to another binary endpoint, which is death. You can see that the purple curve is showing many, many more endpoints than a survival endpoint, which is another endpoint that can be used as a time to failure endpoint. And the power of a discrete endpoint like this is determined by the number of events you can record. And it turns out that if you use this endpoint, this time to first zero, these are, again, I don't want to belabor this table, except to say that you potentially could do a study with a very small number of patients, about 60 patients per group, as compared to about 200 patients per group if you just looked at the changes in slope. So how you look at an endpoint has a great impact. I'm going to just sort of Rudy Giuliani moment. I don't remember, but I'm talking. So electrical encephalography is a measure that in neuromuscular disease does correlate with strength. In the interest of time, I'm going to skip this one and move to sort of the natural endpoint of biomarkers in clinical trials, which are clinically relevant endpoints. And that word has special meaning in the clinical trial world because, in general, the FDA approves drugs on the basis of outcome measures that they consider clinically relevant. And the definition of clinically relevant endpoint is deceptively simple. It's basically a clinically relevant endpoint is a measure of how a patient feels, functions, or survives. That's basically all the entire definition. So it can be completely subjective how patients feel. It can be completely objective, presumably survival is a measure of, and it can be something in the middle. And so obviously survival is a clinically relevant endpoint. Other things which we use very frequently in neurology, such as a clinical rating scale, is also clinically relevant to the extent that that rating scale asks clinically relevant questions. And so, boiled down, it's an easy concept, but arriving at something that is clearly clinically relevant both to you, your patients, and to regulatory agencies is somewhat difficult. And so, an example could be just we were talking about strength. Many neuromuscular diseases have strength as the major aspect of what happens to you over time. In ALS, you get weaker. In myasthenia, you get weaker. In muscular dystrophies, you get weaker. Is strength a clinically relevant endpoint? Well, to date, the FDA has said no, that the result of strength is clinically relevant. If because of weakness you can no longer walk across a room, that's a clinically relevant endpoint. But it's not necessarily true that strength itself is considered to be clinically relevant. One kind of endpoint that clearly is considered to be clinically relevant are functional rating scales. And these are disease-specific. They usually are combinatorial scales, which ask questions about a variety of different aspects of the disease, so that a change in a functional rating scale doesn't necessarily have the same meaning from one study to another, or one part of the scale to another, and I'll show you an example of that. They're often direct assessments where you ask patients about their functional capacity. The amount of change in a functional rating scale that's clinically important rather than clinically relevant is often not completely clear. So that becomes often more of a statistical question than a functional question. And their statistical properties are often misinterpreted, so that most rating scales are discrete scales. Most of the statistics used to describe them and describe changes in them actually treat them as continuous variables. And so there are statistical questions that need to be addressed as well. In general, so I mentioned that they're usually disease-specific. They can be attribute-specific, so that you can have a functional rating scale that just has to do with swallowing, for example, or just has to do with name recognition. Or you can have a rating scale that's much more general. And they are often designed such that you can look at subcomponents and ask individual questions about different attributes of the disease. However, as I mentioned before, they combine different attributes, and so the total change in a functional rate scale doesn't necessarily give you a meaningful measure of how much you've been able to impact the disease. So here's a list of some of the commonly used scales that have been used for approval in the past. Depending on the field you're in, you're going to have different levels of familiarity. One aspect of many scores is that their changes over time are remarkably inconsistent. And so this is the EDSS, which is a commonly used MS scale. And this just shows the number of years in a given patient journey that they will spend at a given EDSS level. And what you can see is that at the beginning of the disease, at low levels of disability, low levels of EDSS, it takes years and years to change from one level to another. In the middle of the disease, it appears that this is sort of the middle of the scale. the changes are much more quickly. Now that could mean that the disease is changing its rate of progression, but more likely what it means is that the attributes that constitutes a grade 4 or grade 5 EDSS are more finely graded. And so the scale itself is producing this change in apparent speed of progression in the disease. So here's an example of a commonly used disease where at some points in the scale, changes could be quite finely graded, but at other points in the scale, the gradation is incredibly gross, and you're likelihood of seeing a change at grades 1 or 2 over the course of a 1-year study. When you spend 3 to 4 years at that grade, the likelihood of that being a useful measure to you is actually quite low. The ALS functional rating scale is a 12-item scale that is divided into 4 different aspects. There are three questions related to bulbar function, 3 attributes related to fine motor function, 3 related to gross motor function, and 3 related to breathing. So an attempt was made to span the entire range of disabilities that can occur in ALS. But just because you have a given number of questions doesn't mean that your scale is accurately sort of amalgamating all these traits together. And so, for example, if you look at the pie graph on the right, what this shows is the changes over time for ALS patients over the course of 6 months in the various domains. And so what you can see is that the purple piece of pie is the respiratory questions, which are a quarter of the questions. But those 3 questions, which are arguably the most important to the patient, only represent 13% of the change over time in an ALS patient's journey. And so what that means is that even though that's an incredibly important attribute, and even though you have the same number of questions asking it, the chance of seeing an effect is actually quite small, as opposed to the fine motor pie area, which is this, which represents 40% of the change, even though it has the same number of questions. So designing a scale isn't simply a matter of saying, well, we have equal numbers of questions asking different things. You actually have to see how fast do these things change over time, what's the sensitivity of each individual question. Finally, a couple of comments about survival, which is a binary endpoint or a time to event endpoint or a time to failure endpoint. In most clinical trials, when you have a survival event, that's a failure. So this is an endpoint that the patient either reach or don't reach at the time of the end of the study. And they're useful, but only to the extent that events occur. I showed you that strength evaluation where if you have more 0 events, you have a better chance of seeing an effect than if you have fewer events. So here are 2 sets of survival curves. These are both in ALS, but one's in a mouse and one is with the phase 3 study of Riluzole in ALS. On the left, you can see that if you look at mice from the beginning of the evaluation period to the end, every mouse has an event. Every mouse has ALS, every mouse dies. And so what you're seeing is the effect of an experimental therapy, which is actually quite subtle. It's about a 10-day survival advantage, but is clearly statistically significant in this study, even though there are only 20 mice per group, because every single mouse had an event. In contrast, the graphs on the right are survival events in the phase 3 study of Riluzole in ALS, which was an 18-month study, where if you look at the end of the study, 60% of the patients are still alive, which means that those patients contributed not at all to your evaluation. They did not have events. And the difference in the number of events between the treated patients and the non-treated patients is incredibly small. So that if you have a pattern like this, you don't need 20 patients per group to see an effect. You need something like 450. So that survival, in many cases, unless you have a very long study or a very highly morbid disease, is not going to be a powerful measure, even though it's definitely clinically relevant. So there you have it. A brief run through a number of things you need to think about when you choose your outcome measures. My view is that even though we only have 30 minutes, this is a critical aspect to designing your trials. Your endpoints not only need to be reliable and meaningful, they have to be sensitive to modification, and they have to be measured in a way that is likely to show a difference should there be a therapeutic effect of your agent. And this is also a really active area of investigation. There are really, I don't think, any aspect of neurology where our endpoints are developed to the point where we can be confident that we've reached the most sensitive way of the trials in terms of measuring the aspects of the disease that we want. So thank you.
Thank you, Dr. Shefner. We have time for a couple questions.
Thank you very much for nice talk. I have a question about like subjective or not subjective, like multi-questionnaire style IPM assessment. I'm a psychiatrist. I'm interested in cognitive decline, for example. Like there's a lot of items. Then, as you pointed out, each item have different trajectory over time. And then, have you seen any examples that based on those trajectories, then some researcher picked one specific one, then focus on those items as an outcome, then publish those data? Have you seen those examples?
So the question has, can you take a complex rating scale and look at some of the subsets of questions and look at the sensitivity of those questions? And that, yes, that's done all the time. And in terms of being useful as a primary outcome measure, that raises questions of what you can't choose what works best at the end of the study. You have to make predictions at the beginning of the study, but. It's not exactly the same, but now that we know that certain muscles change faster in ALS, we're now reducing the number of muscles that we're evaluating and just looking at the muscles that tend to change quickly. The UPDRS, the Parkinson's Rating Scale, has scales that relate to functional activity, to movement, and to cognitive change. Well, the cognitive change subscales don't change over time, in the length of time that is the usual Parkinson's trials. So those questions are often not used in short clinical trials. So people do this, but you can publish post hoc analyses, and they're all very interesting. But in terms of designing a trial, you have to make those decisions ahead of time.
Probably time for one more question, if it exists.
So would it be the quickest changing muscle, or would it be the most significant swallow for the person's life cycle?
So the question of what you would choose, in many ways, it depends on your goal in a given study. In a middle phase study where you're really trying to make a decision of is my drug active and important, you would want to pick the most sensitively changing muscle, most rapidly changing muscle, because most likely that would be the one that would see a change to your intervention. Later on, it's a discussion with regulators and other folks as to what constitutes the importance of your effect. And so it's a much more complex discussion. There are other ways that you can make decisions related to rapidly changing versus slowly changing measures, not just by choosing your endpoint. You can use inclusion criteria for your studies to do that too. So, for example, you might decide if swallowing is most important to you in an ALS study or a myasthenia study, to only pick patients who are changing rapidly with respect to swallowing, and that sort of tilts your metric in a certain way. So you can make the decision based on the intrinsic property of the measure, or about on properties of patients as well.
All right, thanks again, Dr. Shefner.